Good morning, everyone. Welcome here. My name is Xindi from New Jersey Institute of Technology. It's really a great honor to be here to organize this symposium on the brain energy consumption and functional brain organizations. We know that positron emission tomography, or PET, is one of the key neuroimaging techniques that was developed in 1980s and 1990s. And at that time, it is really the first and maybe the only methods that can be used to study brain functions. However, later, of course, we know that functional MRI becomes more predominant due to the fact that there's a lack of radiation exposure and so on. However, PET continue to provide very unique information about neurophysiologies, such as metabolic activity and neurotransmissions and so on. FDG PET, particularly, is a tool, we can use that tool to study glucose metabolisms. And traditionally, usually we use that tool to map the spatial distributions of glucose metabolisms. But recent technique advances enable researchers to study the dynamics of glucose metabolism over time. And also, very interestingly, we can use that to study the relationship between different brain regions that we usually call it molecular connectivity. So today, I'm very honored to invite 4 speakers here to introduce their latest work. Their work usually use FDG PET and combined with some other modalities such as functional MRI and EEG to study the relationship between brain energy consumption and the brain connectivity. And their work shows that highlights the importance of those other modalities that can provide unique and complementary information about our brain functions that can promote our understanding of the brain functions. So today, each talk will last for 15 minutes, and if the talk finishes early, you are welcome to ask questions. Otherwise, I would suggest you to submit your questions via the app. And at the end of the symposium, we will have a discussion session. Okay, without further ado, let me introduce the first speaker, Dr. Bin Wan. Dr. Bin Wan just defended his PhD. Now he's a postdoctoral researcher in the Department of Psychiatry at the University of Geneva in Switzerland. Today, his talk is entitled Bridging Glucose Metabolism and the Intrinsic Functional Organizations of the Human Cortex. Thank you.
Hello. Thanks for the introduction. I'm so happy to be here today to have my talk about bridging cortical glucose metabolism and intrinsic functional organization. Yes, I just defended my thesis last week, and I'm finishing my PhD. I'm also starting my postdoc at Geneva. How can I do?
You just say next slide, so he will start, but there will be a slight delay.
Okay, so next slide. Sorry?
Sorry, this one. This one.
Yeah, sure. So I will stay 10 seconds for this to scan the QR code to see my preprint a little bit if you want to know more. Yeah. I have no disclosures. And we all know the brain is a really energy-consuming organ in our body. It consumes 20% energy of the whole body. And the neural activities consume about like 70% energy of the whole brain. And we know the brain contains 9 neurons and non-neural cells. And for the neuron cells, it mostly depends on the oxygen-related energy consumption. And for non-neural cells, and some of the cells can use the non-oxygen usage pathway. And for the energy consumption in the neurons, the mitochondria is a really important, what is called like organ or something. So if there is like a mitochondrial dysfunction is in the brain, then it will cause a lot of like brain-related disease, like for example, AD, autism or schizophrenia. But however, how the brain energy and function association is how the brain function and energy, the relationship is not really fully understood. And yes, then a really important tool is like PET. It can measure the in vivo regional distribution of the energy consumption in the brain. Here, a general principle of the PET fMG imaging shows the frontal and visual areas, they show a high demanding of glucose consumption, but however, the sensory motor areas and like parietal and anterior temporal cortex, they show a low demanding, but there is still like individual difference, and this is a general pattern. The correlation between the glucose metabolism and the mitochondrial map, there is a not high correlation, it's like 0.2. So it can explain that brain not only rely on like oxygen reactions, but also like non-oxygen reactions. And yeah, so we want to understand how the brain energy can inform the brain function. And some previous studies, they use such as like topological metrics, such as like Radholm, the local activities, and degree centrality, and FALF. And they find all these metrics can explain like 20 to 30% of the variance of the PET-FDG. And yeah, this is kind of like moderate correlation, but however, we want to understand more what kind of feature of fMRI should be best to use to fit the FDG map. And if we jump out of the like topological graph of the functional collectome, what we can do. And here, if we look at this map, the FDG PET map, the color more closer to each other, the regions, if we think they show the similar energy consumption, then maybe they have similar functional collectivity profiles, and we ask these questions. So conceptually, difference between the regional functional collectivity would reflect the spatial difference of glucose demanding, and similar functional collectivity profiles could drive a similar glucose demanding. And if yes, then from the methodology, the low-dimensional gradients are from the functional collectome similarity cortex can reconstruct the FDG map if it's true. So, and there is our motivation to use the functional similarity to fit the FDG map is from this year's preprint. If you want to look at, like here is a QR code for the GWAS study. And can I go last slide? Yeah. So here we use the UK Biobank data individual to fit the HCP template and calculate the global feature of the functional gradients. And then we perform GWAS on the global feature, and we find 5 loci. And these 5 loci is enriched in the metabolic pathways. And when we expand these pathways, we find the glucose-related and lipids-related pathways are really important for the shaping of the functional gradients and for the biological reasons. And we want to study the functional gradients, how it can reconstruct the FDG PET map. Yeah, here is a QR code if you want to know more about this preprint. And this is our final framework to go. Here we show all the background of the glucose metabolism in the brain and how the oxygen related, and then we measure the oxygen by fMRI, and then we construct the functional collectivity profiles. If it's similar, then the region A and region B show a similar energy consumption, and we use the no dimensionality gradients, the x-axis, to reconstruct the FDG PET map. And here our first results showing we use the 20 individuals, and here we show a group level results. We calculate the group level functional collectivity matrix, and then sparse the matrix and get the affinity matrix to calculate the row-wise correlations. And then we use diffusion map embedding to calculate the no-dimensional gradients of the matrix. And here we use different parameters like top 10%, like sparsity equals to 0.9 to calculate the no-dimensionality. And yeah, here we show a group brain patterns of the sparsity equals to 0.9, which is like 10% of the functional connectivities. And we show here the G1 the first axis captures the unimodal to transmodal gradients, and G2 captures the sensory to visual gradients, and as well as like G3, and we calculate 100 gradients to finally to fit the FDG map. Here we use a stepwise regression, and inspired James Pan's Nature paper in 2023, and use eigenmodes to fit any brain functional map. And here we show with more gradients added in the model, the performance just grow, and the convergence is like stop at 60 gradients. And when we use 5 gradients, the fitted map and the correlation between the fitted map and the empirical map is like a correlation is 0.5. Is such really comparable to the previous findings using the topology graph. And when we use 60 gradients, they are reached to 0.8. Yeah, and because we use a lot of gradients, access so many brain maps, and some people may ask if you use like any like 100 or more than 50 different brain maps, you can fit any brain map. So here we use spatial autocorrelations to generate the 100 surrogate maps using the geodistance matrix based on brain smash, if you know this toolbox. And we first permutate the glucose metabolism map for 100 times, for 1,000 times. And we see here the empirical map, the empirical fitting is greater than 996 surrogate maps. And when we permutate the gradient maps for 1,000 times, we find the empirical map is greater than 1,000 surrogate maps. And this is all based on group level results, and we want to find whether this kind of fitting can be generalized to different individuals. So here we also perform individual fitting, and we use the raw gradients. If you guys know like the approach of gradients, we usually use the aligned gradients to make individual comparable to do like case control comparisons. But here we want to generalize this result, so we use raw functional gradients here, and we show 2 subjects here as S003 and S020, and they really show different gradient maps. However, the fitting trend is still like growing and shows growing and growing, and the convergence is like also like 60 for different subjects. But however, there is a huge like individual variation for the fitting, but the general average fitting is like variance explains like mean is 44%, and yeah, and we think gradients might be the general principle of the glucose consumption, but the inter-individual variabilities, why there is so large individual variation, it may be correlated with the mitochondrial differences in the different regions and different persons, but we can not test it because mitochondria is really from like that person, and we cannot use in vivo data to fit it. The second reason we think maybe different modalities, like if you include structure like James Pan's eigenmodes, may can increase it or not, and whatever we do this, and finally it may also related to different mental health status of different subjects, but here we all use like healthy individuals basically, but we don't have like mental health scores for them. We cannot test for now, but for the different modalities, we test the geometric distance eigenmodes, and here functional connectivity gradients, and we also use geometric distance eigenmodes plus functional gradients, and we see here the geo-distance eigenmodes have a large individual variation, and the fitting is kind of lower than functional gradients. And here, every curve is every individual. And finally, we use the geo-distance plus functional collectivity matrix. We find the inter-individual variation gets smaller, but the performance is kind of similar to the functional gradients. And the last results I want to show here, because my PhD thesis is based on brain asymmetry, and this is my fourth project in my PhD, and I want to study the brain asymmetry, how it relates to the, or brain functional asymmetry relates to the brain energy consumption asymmetry, based on the theory like the brain has two hemispheres of the brain, just want to avoid a duplication of the neural circuits, so it can display the difference between the hemispheres. And here we use the functional gradients asymmetry to fit the glucose asymmetry. We propose like 4 modes here. And first we have to fit the FDG pad and functional gradients in different hemispheres in like panel C shows here. And the 5 modes we propose here is the mode 1, the functional gradients regression coefficient used for the left and left, and right for right, and then exchange left and right for the, if the left hemisphere and the right hemisphere, they don't share the regression coefficients of the energy. If they share the energy consumption coefficients, then 3 models. So the model 3 just uses both hemispheres use left hemisphere coefficients, and the model 4 both hemispheres use right. So they use right hemisphere coefficients, and the model 5 uses the asymmetry coefficients. And finally, we find the model 1 is greater than model 5, and the model 3, and model 1, model 3, and the model 2, model 1, model 2, they belong to the brain asymmetry, the series competition. Just as I said, the two hemispheres using different energy-related regression coefficients to fit the from function to energy. And the other 3 modes belongs to the lateralization mode. And this concept is really, comparison concepts is really novel. And if you want to discuss more, we can discuss like in person. And yeah. And then we find final conclusion is competition works better than lateralization for brain asymmetry. And to conclude, we find regional difference of glucose metabolism can be effectively built on the interregional functional collectivity similarity. And these reconstructions are relied on their own maps instead of like random maps. And when we use 5 gradients of the functional collectome, it is really comparable to previous findings. When we use 60 gradients, then the explanation could be greater than 70%. And there exist individual differences, might be insightful for mental health to observe fMRI glucose coupling. It is also necessary to input the structure constructs to test the findings. And then finally, we find 2 hemispheres may prefer to use different energy regression coefficients to work rather than under a uniform theory to assign them to tasks. Yeah, I want to thank all the collaborators, and if you want to know more about this work, you can scan the QR code on bioRxiv. Thanks.